The overall goal of this procedure is to prepare samples of membrane protein crystals in lipidic cubic phase for serial crystallography data collection at synchrotron and X-ray free electron laser sources.
This method should help answering key questions in the field of structural biology of membrane proteins, such as transport and single transduction across the membrane.
The main advantage of this technique is that it enables structure determination of membrane proteins in their native lipid environment at room temperature with minimal sample consumptions and negligible radiation damage.
Individuals new to this method will struggle because lipidic cubic phase is a viscous gel-like material that is difficult to handle without special tools and prior experience.
Begin this procedure with the reconstitution of membrane proteins in the lipidic cubic phase, or LCP, using syringes number 1 and number 2 as described in the text protocol. After a transparent and homogeneous LCP is formed, move the entire sample into syringe number 2. Detach syringe number 1 while keeping the coupler connected to syringe number 2. Connect a removable needle to another 100 microliter clean syringe, called syringe number 3. Aspirate approximately 70 microliters of the precipitant solution into it. Disconnect the needle from syringe number 3 while keeping the Teflon ferrule inside the syringe. Connect syringes number 2 and number 3 through the coupler, making sure that Teflon ferrules are correctly in place in both syringes. Carefully screw the coupler tightly into position. Orient the coupled syringes vertically.